Hello everyone. Today I am going to discuss about cloning and gene manipulation. So this is the last topic of this module, and here particularly I will be discussing about clone and the cloning method, overall idea about these two terms, followed by cloning whole animals. And there are two major methods here. One is embryo splitting method and the other one is nuclear transfer method, followed by gene manipulation or gene editing. So here the first point I am going to discuss about clone and cloning. So clone means organisms carrying identical genes, so that we will say those two are clone to each other. During the last lecture, what I discussed, that is mostly about cloning of a gene. So like if I summarize that, what I mentioned there, that you have one plasmid. So this is plasmid, and here you have one gene of interest. I would say if this is your gene of interest, so this is gene of interest. Now, if you digest this gene of interest by two restriction enzymes, for example, I would say ECO R1 and here BAM H1. So these are restriction enzymes which I discussed during the last lecture. Similarly, in plasmid you have many important properties, and you have some portion which is called multiple cloning site. This is called multiple cloning site, MCS. So you have here many different choices of restriction enzyme. So as a result of that, you can digest this plasmid by the same set of enzyme, that is ECO R1 and BAM H1. So as a result of that, your plasmid will be in a linearized form. So I would say you might get something like this structure. So this is now your plasmid after digestion with ECO R1 and BAM H1. Similarly, from your gene of interest, you will be getting something like this. So this is your gene of interest. Now, since we use the same set of enzymes, so this end here and this end, they can base pair here, they will be complementary to each other in this region. So as a result of that, you can actually put your gene of interest here in this plasmid, right? Now, on the basis of selection and all those things, what you have to do, you have to transform some host cell, for example bacteria, you are sending this plasmid, and then when the bacteria will divide into many numbers, then this plasmid will also grow in number, and also plasmid can replicate by itself. So as a result of that, many plasmid will be there in one bacteria. So now as a result of that, what you are doing, you are increasing the number of your gene of interest. So those are exactly same gene. So those are the cloning of gene. So your genes are now cloned. So you have multiple copies of same gene by this method. So this is cloning of genes. Cloning of gene. So this is just I summarize whatever we discussed before. Now here I mention in context to some organism, that organism carrying identical genes. So this cloning method is very, I would say, primitive if you think about plant system. So all of us, we know that, for example, if this is a plant, right? This is just plant, and okay, plant. Many plant, if you cut this portion, one branch or one stem, and you put it in suitable environment in soil, and you are pouring water, everything fine, then you will be seeing that you can have a new plant here, right? So new plant here from this portion only. So this is cutting method. So now if you see in terms of genetic, like constituent of these two plant A and plant B, so you will be saying they have exactly same genomic material, because we are just putting one branch, or we are cutting little bit of this plant and this plant is growing again. So this is very basic method of, I would say, cloning in case of plant, just by cutting. And more advanced methods are, for example, tissue culture from like plant cell, particularly plant tissue, you can, in a laboratory environment, you can grow in in number like plant cells, and then in control environment with proper nutrients from that tissue, you can have multiple same plant. So as a result of that, those are also cloned to each other. They are exactly same plant. But naturally, what we generally see, for example, if I would say this plant A, if this is flowering plant, for example, then after plant will be matured, then flowers will be there, some pollination will be happening, and some fruit followed by seed and some new plant. That is not the clone, because during pollination, meiosis happen, like to make the ovule as well as the pollen, and as a result of that, the exact genetic composition is not like the parent plant. So as a result of that, those are not cloned. But whatever I discuss right now, by cutting method or by tissue culture method, you can have plant clone also, right? There are several methods. I am not going to discuss that. So now this is the cloning thing in context to organism I just mentioned, and also I mentioned about cloning of a gene. Then cloning in animal, this is little bit, I would say, problematic, because not like plant tissue, animal tissue will not always give you it cannot grow properly, right? Only stem cells, they have the power, like they are totipotent, so you can probably make a new animal from stem cell itself, particularly I would say embryonic stem cell when it is zygote, or just at the very early division, during that stage you can make that. Now here cloning of animals I will discuss in this point separately, like I will emphasize here. Like one method is called embryo splitting. So in this case, what we will see here, that for example we are taking cell. So this is I would say, if I consider this is the female shape and this is the male shape. So now from this female shape, egg can be collected. This is egg cell. And similarly, from this male shape, sperm can be collected. And now what will happen if we conduct IVF, that is in vitro fertilization? So as a result of that, you will get zygote, right? And then in laboratory environment, because you are doing in vitro fertilization here, so still it is in lab. Now this zygote will be divided into a few cell stage, I would say 8 to 16 cells stage. So those are still, like every cells, they have power to make anything in the body. So they are properly embryonic stem cell. So now if surgically we split these cells into two groups, right? Surgically we are separating this cell mass. So cells are separated. And now these separated cells, we can put it in surrogate mother. Surrogate mother. So it's another actually shape, surrogate mother. So you can put this one into another surrogate mother or the same surrogate mother, that doesn't matter. But finally, what you will be getting here, like this surrogate mother will give birth to these shapes, right? Now, these two shapes, they are identical to each other. They are identical because both of them, they are actually originated from this zygote, the same zygote. So, they are not identical to their parent, like here this female sheep or male sheep, they are not like that. But these two, they are clone to each other because they are originated from the same zygote. So, this is the embryo splitting method, and by which you can get this identical shape, and these two are clones. And these things sometimes happen naturally in our human population also, not in this way, but naturally. So, as a result of that, what I would say, after fertilization, the zygote, when it is getting divided at very early stage, accidentally by some other reason, if it divided into two, and both of those two set of very early embryo, if they are implanted in the uterus, then that mother will give birth to identical twin, right? So, they will be exactly identical. So, identical twin, because they are coming from same zygote. But in case of non-identical twin, that's a completely different scenario, where two different egg cells, they are fertilized by two different sperm. So, in human population, you will be seeing both, sometimes identical twins, sometimes non-identical twin. But I hope that you understand the basic difference between these two set of twins. Now, we will be discussing the next technique here, cloning by nuclear transfer. So, this nuclear transfer, particularly this is also known as somatic cell nuclear transfer. So, SCNT, somatic cell nuclear transfer. So, here for example, like many of us, we know that the first cloned mammal is Dolly, one sheep actually, right. So, the same way which I am going to discuss, Dolly was generated in the same way. So, here what we do, so as you can see, this one, this is I would say shape A, and this is shape B. So, in this case both sheep can be female, no problem. It can be one male, one female, that is also okay, but one female is required because we will take one egg cell. I will be discussing why. So, now the thing is, if I say sheep A and sheep B both are female sheep, for example, now we are taking some soft tissue from this sheep, soft tissue, so that we can quickly take the cells and properly we can handle it, just for experimental reason, some soft tissue, and from there you will get a cell. What kind of cell? Not egg cell, not sperm cell, this is somatic cell. Somatic cell, that means any body cell. So, therefore, it will contain the whole genetic information. As you know that sperm and ovum, egg, they have half of the information, right. But in this case, the total set of chromosomes should be there, like diploid number of chromosomes should be there in this somatic cell. So, I would say if this is the somatic cell here, this is the somatic cell, and at the center in black color, whatever I am showing, this is the nucleus. So, I would say this is nucleus. So, we will take this nucleus out, right. So, nucleus was removed and it will be used in this method, and the cells without the nucleus, it will be discarded, no requirement. Similarly, from sheep B, as I already mentioned, this is a female sheep. So, from this sheep, we will be getting the egg cell. So, this is egg cell. Here you have half of the chromosome, but that does not matter because we are not relying on the chromosome here, like egg chromosome here. So, we will be like removing this nucleus. So, just remove the nucleus. Nucleus is not required here. Remove the nucleus. Just take the empty egg. Empty egg. And now we will be fusing this together. This nucleus from the somatic cell, which contain the whole information, and the empty egg together. So, see, this is not the fertilization like sperm and egg are getting united. We are putting the full nucleus from the somatic cell inside the egg. That's all. And then here you need little bit of strategy, some little bit of electric pulse and all those things, and everything is happening in lab. And then this one will start dividing, and it will make like early embryo, few more cells will be there. As you can see, like early embryonic stage here. And this one we can transfer it into surrogate mother. So, this is here surrogate mother. So, now as you can see, this is embryo, and if we put in surrogate mother this embryo, then she will give birth to a baby sheep. So, this is baby sheep. So, now if you see this sheep, the baby sheep exactly looking like which one? Neither like sheep B nor like its surrogate mother. The sheep is exactly similar to the sheep A, just like sheep A. Why? Because the nucleus we have taken from sheep A. So, this process is called somatic cell nuclear transfer for the cloning of whole animal, right. Now, this empty egg is absolutely required, because inside the egg, apart from the nucleus, there are many important ingredients, particularly many mRNA and proteins which are absolutely required, and that is why we have to take the empty egg and we are fusing with the nucleus, and finally we are getting this clone sheep. Now, here this baby sheep, this one, and sheep A, they are cloned to each other, they are same thing, right, they have exactly same genetic composition. So, now by this method Dolly was created. As I already mentioned, Dolly is the first cloned mammal, and it was created and I would say it born on 5th July in 1996 at Roslin Institute at Scotland, and the sheep was overall healthy, and it has also multiple baby, and at around 6.5 years age, actually it showed some kind of problem, and finally it passed away. So, this is whatever I discussed, two different method of whole animal cloning. Now, apart from those cloning, like whatever I just discussed, some interesting facts are there. I am just mentioning those for I would say for fun. So, if you see here, in 2001, scientists at Texas University, Texas A&M, they have created the first clone cat, right, by the same method, that somatic cell nuclear transfer, the same method. So, this is known as like clone cat, but they also like this is copycat, because this is same thing, right. So, copycat. And here if you see, this is I am taking this image from this issue of Nature journal. So, here from this cat, somatic cell nucleus was taken, and this is the surrogate mother, and this is the clone cat. Clone cat. It looks like cat A here. So, this is similar to cat A because it is clone cat. And if we move little bit further, whatever I mentioned about some example of cloning, Dolly is very famous, like all of us we know, but the cat we might not know. But this one, this is little bit more interesting because this is the first cloned primate. So, primate means it's very close to human. This is monkey. This is particularly called crab eating monkey. These are small in size, and their name, if you see, Zhong Zhong and Hua Hua, they born in 2017 November in the Institute of Neuroscience of Chinese Academy of Science in Shanghai. So, the method is same, like somatic cell nuclear transfer, and they are also healthy and they are okay. So, as a result of that, if you see the overall, the advancement in technology and understanding in science, those are, working fine. But now the question is coming about ethical issue, what we should do, what we shouldn't do. That is more important question here. And also I would like to mention here that whenever I was discussing right now about the cloning of whole animal, whether it is by embryo splitting or nuclear transfer, you will be noticing that here we are not really like cleaving some particular DNA and then again we are integrating DNA, not like that. This is like, I would say, we are not relying on any those kind of enzymes like ligase, restriction enzyme, not like that. This is more like kind of natural process we are trying to mimicking, right? Now we will go little further development of what is happening now. So this one particularly, this is again something interesting fact here. So if you see, this is one Hollywood movie, and the name of the movie here is Gattaca, like Gattaca. So now there is no language like no word like Gattaca here. So this is nothing but nucleotide sequence. Now all of you know, right? It's ATGC sequence present here. So the name of the movie itself is GATTACA. This is just nucleotide sequence, and this is a science fiction movie, and this is, I would say, mostly talking about selecting best possible features from parents, and this is, I would say, very enjoyable movie, particularly if you have little bit knowledge about genes and genetic engineering, all those things, and which you already have, so you can enjoy this. And now here, this is little bit some controversial topic here, that Lulu and Nana. So these two are actually human baby, but this is gene edited human baby. That's why I just few minutes back I was telling that we have to be very careful about which is ethical and which is non-ethical. So in this case, what we came to know, so for example, this is from BBC News, that gene editing babies called to pause human altering research. And here particularly I have taken this from Science magazine here. So did CRISPR help or harm the first ever gene edited babies? So as a result of that, there are a lot of concern also. So CRISPR means some kind of particular technique which makes everything like very easy to edit genes or genomes even in human, as you can see here. So I will be discussing about CRISPR in the next slide. But the problem here is what happened, that this scientist, he in China, so he edited like the genomes of these two babies, Lulu and Nana. So that's why all these concerns coming. So whether it is good, bad, and it has some long-term effect and some ethical issues also. So like as I mentioned, the scientific community should be very careful about this thing, and which they are rightly doing. And now if you see what is CRISPR-Cas, technology. This is very important, and this is very robust technique. Nowadays, almost many of us, we are using this in day-to-day basis in lab, that particular this technique CRISPR, which was used here also in this case. So particularly I would like to mention for this technique, it is discovered, this technique is very, I would say, new technique. So this is only discovered in around 2012 to 15 during that time. And for this discovery, Emmanuel here and Jennifer Doudna, so they received Nobel Prize in 2020 in chemistry. So they got Nobel Prize in chemistry in 2020 for their gene editing technique based on CRISPR, based system, CRISPR-Cas based system. So what is CRISPR-Cas system? That I would like to highlight. I would not go, much more details, but I will introduce that idea. The thing is, CRISPR-Cas system here, this is very big name, CRISPR. So if you see here, what is the meaning of CRISPR? That is clustered, regulatory, interspersed, short, palindromic repeat. Very big name. If you forget, nothing will happen. I will discuss, I will give you the idea of what is CRISPR. And CRISPR-Cas system. So Cas means here CRISPR associated protein. So CRISPR associated protein. So this is Cas system. Okay. So together this CRISPR-Cas can be utilized in a very robust method, and it is very useful nowadays even for academic research. Like very frequently and effectively we can edit genes for our academic research, for example, like to knock out, to delete some genes from some genome, or to introduce some new segment of a gene by this technique. So let's see what is this technique. The idea is, again, this is coming from some natural observation. During the last class, I mentioned about restriction enzymes, and I particularly mentioned that restriction enzymes were isolated, purified from bacterial system. Bacteria, they do use restriction enzyme for their defense mechanism. They use that to degrade, to destroy phage virus DNA or bacteriophage DNA. So this is, there I would say some kind of immune system, but that is innate immune system. But here in this, like CRISPR based system, this is also some kind of bacterial immune system, but this is adaptive immune system. So like Emmanuel particularly, she learned a lot of those things from bacterial system, the CRISPR system, and together they modify the system so that it can be more applicable for our own purpose. But the bottom line is, you can see this bacteria, they naturally, they evolved, they generated this kind of system by themselves to get protection against phage virus. As I already mentioned in the first lecture, the lecture 1, that in living system, because of evolution, most of the organism, they solve most of the problem also. So as a result of that, you can see bacteria, they solve problem of their bacteriophage infection. Sometimes they have restriction enzyme to degrade foreign DNA, but they are protecting their own DNA. Similarly, here in CRISPR system, this is also against phage virus DNA, but this is some kind of adaptive immune system. Let's see what is happening here. So I would say, if this is bacteria, inside the bacteria I am showing here, this is bacteria. Now phage virus, you can think about T2 phage, for example. This is phage virus. So what they do? They inject their genomic material, phage DNA I would say, they are injecting inside bacteria, so that more phage virus can be generated and the bacteria will be lysed and more phage particle will be generated. But the thing is, bacteria is trying to protect themselves by some kind of method. So what is happening here? So in bacterial chromosome, like what scientists observe, they have some kind of repeat, like this CRISPR-Cas system is present inside bacterial chromosome itself. What is that? So I would say, if this is bacterial chromosome, so this is bacterial DNA, bacterial chromosome, same thing. And I'm just, zooming little bit of this portion, so where all these things are happening. This is part of the bacterial chromosome itself. So what is present here? You have some portion, so this is called Cas genes. So Cas genes, this is already present in bacterial DNA or bacterial chromosome. Followed by, you have CRISPR array. So what is CRISPR array? So you have some repeat and followed by some spacer. So I will explain that. So I would say here, particularly if I say this is repeat, 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 right? So this is some kind of short palindromic repeat. And in between you have, like for example, this one, this is one particular DNA sequence present here, okay? So now the thing is, those are actually acquired by bacteria. During phage virus infection, they are acquiring those things. So this is nothing but phage DNA. This is nothing but phage DNA, not the whole DNA, segment of phage DNA. Why? As I already mentioned, this is just like adaptive immune system. What is happening? For example, this phage virus, when infecting, it has some specific region, and I'm not going into that much of details, but that during this infection cycle, this should be incorporated here. So for example, this is the same thing here. The phage virus DNA is getting incorporated in this small segment. If I say this is phage A, a particular name here, and this is the A region, so it is integrated here. So together I would say this is CRISP array. Now what will happen? This CRISPR array, it will be transcribed into mRNA, that is called pre-CRISPR RNA. And some more modification, there are many enzymes involved also, then you will get CRISPR RNA. So why I am making small fragment? Because you are getting one, like this is 1, 2, 3, for example, from this portion, 1, 2 and 3. And Cas genes, after transcription, translation, it will make Cas proteins. And Cas9 is one of those important proteins. Cas1, Cas2, those are also very important, but Cas9 is also very important. So now Cas9, they will form complex with this CRISPR RNA. So for example, I would say, if they form some kind of complex like Cas9 and this CRISPR RNA, particularly CRISPR RNA 1. So this is some kind of complex, Cas9 protein and RNA itself. So what will happen? This complex will help to prevent this bacteria for further infection by the same virus. So what will happen? Whenever now the same virus will infect this bacteria, the same DNA it will receive. As a result of that, you have this CRISPR RNA, which I label by 1, and now this CRISPR RNA will actually recognize the complementary sequence which is coming from the viral DNA. So as a result of that, this CRISPR RNA will bind to the viral DNA, and now this Cas9 protein will act on it, and as a result of that, you will be seeing the viral DNA will be destroyed, degraded, like double stranded break will happen. In this way, they are actually preventing themselves from phage virus infection. Initially, they will be infected, and during the infection, they are acquiring this short segment of the DNA. But if some new virus infect this bacteria, it has no this signature motif in its DNA itself. So during that time, it will go through the infection cycle, and then it will acquire a small segment, and it will be integrated with the CRISPR array. But next time, if again the same virus infect, that time the same, like I would say, the CRISPR RNA will be generated, and along with CRISPR RNA and the Cas9 protein, the viral DNA will be destroyed. This is the overall technique, how bacteria, they prevent themselves. This is very, I would say, similar to mammalian adaptive immune system, but by their own way. And this was like extensively characterized by Emmanuel and also by Jennifer, like, they like particularly make it more convenient. Instead of CRISPR RNA, they design some like, guide RNA so that it is more programmable. It can be used very, in a easy way. And nowadays, what we are doing, we are using Cas9 and some guide RNA. So together, like you can put that in some plasmid, and then you can actually send them to your, particular cell where you want to modify your genome. So that is very common nowadays, to delete some portion of gene or to add some extra segment. You can do a lot of those kind of, gene editing technique. So here you can see this is the guide RNA, and this is the Cas9 protein, like, this all in blue color, this is the Cas9 protein, and this is the genomic DNA where you are actually, trying to edit it. So this is kind of, brief introduction to CRISPR-Cas system, but as I already mentioned, this is very useful, and very quickly they received Nobel Prize also in 2020 because of their, this like very important and interesting work. And that's all. So thank you very much.